I'm Louisa Scott.
And I'm Sangeeta Iyer.
Our research at the University of Texas at Austin focuses on finding new drug candidates for diseases like Alzheimer's disease. Sangeeta will tell you about the Alzheimer's disease project.
I and others in the Pierce Shimomura laboratory study human diseases using a 1 millimeter long worm called C. elegans. What may surprise you is that this tiny worm shares many similar genes and proteins with humans. The genetic material of worms can be manipulated to make the worm churn out virtually any foreign protein in cells of our choosing. We have used this technique to make worms mimic features of Alzheimer's disease. Alzheimer's disease, or AD, is characterized by the accumulation of certain proteins in the brain and subsequent loss of brain cells called neurons. Different areas of the brain control different functions. The accumulation and spread of abnormal protein throughout the brain results in the many debilitating effects of Alzheimer's disease. To model this disease in worms, we must first replicate the abundance of the abnormal AD protein. Second, the corresponding death of the neurons, and ultimately the change in behaviors previously controlled by the dead neurons. The Alzheimer's disease worm bears all of these hallmarks, making it a useful model to study this disease. Cholinergic neurons are a class of brain cells that are particularly susceptible to death in people with Alzheimer's disease. Here, you can see a worm with all of its cholinergic neurons producing a green fluorescent protein. We engineered these worms such that a set of these cholinergic neurons would also produce a protein that accumulates in AD, the human amyloid precursor protein. In this panel, 2 of the cholinergic neurons are producing green fluorescent protein. In the panel below, you can see that the 2 neurons also produce the human amyloid precursor protein indicated by red fluorescence. In these worms, cholinergic neurons die much like they do in people with AD. Here, you can see healthy cholinergic neurons in a young animal. But these neurons are missing in an older worm producing abnormal AD protein. The cholinergic neurons that are affected in the worm control swimming, which normally looks like this. Worms with dead neurons swim poorly, like these worms on the right. We are searching for new AD treatments by studying the effects of known and novel drugs on our worms with AD symptoms. We want to find drugs that keep the cholinergic neurons healthy and the worms behaving normally. Already, we have found some promising drugs that slow the progression of AD in the worm.
You can help us expand our research to test more drugs. Your contributions will help us purchase equipment to continue researching new treatments for Alzheimer's disease.
Thank you for watching.